Okay, I have to
All right.
present.
Hello, everybody. Good morning to everybody who is on the East Coast, and to everybody else, good afternoon or good evening. So we have the pleasure today to hear a talk by a subject that many of us became interested in the last few years, but we don't have that much experience, so we need the specialist in the area to tell us what's going on in the subterranean estuary in terms of microbial dynamics and microbial changes. And it's quite important, of course, because those microbial communities drive the geochemical changes. So our research becomes biogeochemistry rather. So Clara González will tell us about her work, and yeah, I'm giving to Clara.
Okay. So good afternoon. Thank you everybody for coming, and thanks Natasha for this invitation. So my name is and I work at the Institute of Marine Sciences in Barcelona, and today I would like to tell you about a review paper on the microbial dimension of submarine groundwater discharge, or SGD, that I published last year together with Valentí Rodellas and Jordi García Orellana from the Universidad Autónoma de Barcelona, also in Spain. But first, I would like to start by saying that I am not an SGD expert. As you can see from this drawing that represents how I had imagined submarine groundwater discharge when I first started hearing about it when working with Valentí and Jordi. So I am instead aquatic microbial ecologist, and I started to work in this field during my PhD thesis in Barcelona at the Institute of Marine Sciences, where I studied marine bacteria and their responses to different factors. But then I moved the focus of my research to freshwater ecosystems, first at the University of Girona, also in Spain, working with bacteria from rivers, and then during 3 years in Canada, where I studied bacterial communities from freshwater and terrestrial boreal ecosystems. So when I came back to the ICM and therefore to the study of the ocean again, I wanted to keep this link with the terrestrial and freshwater ecosystems as part of my research. So the study of submarine groundwater discharge seemed like the perfect way to keep working at the interface between these freshwater and marine worlds. And actually, the research that I did during my time in Canada was what motivated actually my current interest in submarine groundwater discharge, and I'll tell you why. So during this time, I studied the biogeography and the ecology of bacterial communities from these complex boreal landscapes where the freshwater and terrestrial environments are very tightly connected. And what we found was that we cannot understand the freshwater microbiome without considering the terrestrial linkages, the linkages with the surrounding terrestrial environment, because we found that lake and river boreal microbial communities received large amounts of microorganisms and dissolved organic carbon from the surrounding soils, which largely influence the structure and function of these communities. So this may seem obvious because we all know that freshwater, terrestrial, and marine ecosystems are very tightly linked in nature due to the movement, mostly to the movement of water in the landscape. But the truth is that most microbial ecology studies have been based on individual types of ecosystems, such as only streams, only in lakes, only the ocean, thus disregarding this natural connectivity between habitats. And actually, this marine interface connected by groundwater is one of the least studied habitats in microbial ecology. So likewise in boreal lakes and rivers, the marine microbiome is also, oh sorry, there's somebody calling. So it's also, the marine microbiome is also subject to some extent to these linkages with the surrounding terrestrial environment, one of which is the so-called submarine groundwater discharge, or SGD, as you all know. So for example, in the Mediterranean Sea, where we are mostly working, it was estimated that the nutrient inputs associated to submarine groundwater discharge are equal or higher than those from rivers or atmospheric deposition. So given the semi-enclosed and oligotrophic nature of the sea, this hydrologic, sorry about that, this hydrologic process will have large impacts on the marine system. Okay. So a few years ago, I started to work with Valentí and Jordi, trying to sample some of their most frequently visited coastal aquifers. And when I finally got a project to work on the topic, then the COVID came. So we could not sample anymore. But we decided to use the confinement time to work on a review on the topic of the microbial aspects associated to submarine groundwater discharge. So today I will present you some of the highlights of this paper that was published last year. But also almost at the same time, there was another review on the same topic from Arcana and collaborators that also provides a nice overview on the topic, although more focused on the beach aquifers. So I also encourage you to check it. So, okay, what is this microbial dimension of SGD, of submarine groundwater discharge? So first we have the role that microbes play in the chemical transformations of groundwater within the aquifer or along the subterranean estuaries that are believed to determine or at least modulate the chemical fluxes associated to SGD. But also these water, chemical and microbial inputs associated to SGD will have an effect on marine microbial communities with potential ecological consequences. So today, so we could say that microbes control the flow and fate of groundwater elements into the marine environment, but the truth is that we know very little about these communities at the terrestrial marine interface and about how these communities may modulate or affect the effects of submarine groundwater discharge in the ocean. So today I will talk about this, but first I would tell you very quickly about how we can study natural microbial communities so that more or less everybody is on the same page. And I work mostly on bacteria, so I will focus on these microorganisms. So we can do different things. We can, for example, try to isolate and culture some bacterial species in different culturing media, like these agar plates, so that we can experiment with different bacterial species, learn about their physiology. So this is really useful, but we know that most bacterial species cannot be cultivated. So this can only be applied for a very small fraction of the natural microbial diversity. We can also observe microorganisms with microscopy so that we can estimate their numbers, visualize their morphology and size, or quantify their biomass, for example. And finally, we can try to identify bacterial or microbial species and functions through the sequencing of their genetic material. And you may have heard that actually the field, this advent of sequencing techniques is what has revolutionized this field of microbial ecology, because they allowed an unprecedented description or characterization of the microbial diversity. So very briefly, these techniques could be divided into the detection by PCR of specific marker genes that can allow identifying microbial species in general or specific functional groups, such as, for example, the nitrifiers, or other more recently developed techniques such as metagenomics or metatranscriptomics that allow the sequencing of all the genetic material of communities and therefore provide an idea of the metabolic potential of a community or the metabolic diversity within it. And in turn, this genetic information can be coupled to microscopy techniques to identify specific microbial groups, for example, or to gain genomic, more insight on the genomic information of bacterial isolates of interest. So in our review, we searched for papers published between the years 2000 and 2020 that contain the terms groundwater and microbial in the title, and found this large increase in the number of studies that was clearly linked to this advent of sequencing technologies. However, when we included terms such as submarine, seawater, coastal aquifer, or subterranean estuaries, we retrieve much less papers. So this indicates that most of this research has been based on inland groundwaters, and therefore that coastal aquifers had received comparably less attention. But what is it that makes groundwater so interesting from a microbial point of view? Okay, so first, aquifers have been much less sampled than know much less in general, but they are believed to hide a large fraction of the total global bacterial biomass. So for example, in this study, they estimated that roughly the 90% of the total bacterial and archaeal biomass is contained within subsurface ecosystems, including aquifers and sites below the seafloor. But more interestingly, they hide an enormous taxonomic and metabolic diversity and novelty. So to illustrate this, here you have a tree of life where you can see that the diversification within bacteria is huge compared to that of plants or animals. So, but if you see here, this whole branch indicated in purple, so this whole branch of new groups was discovered in groundwaters less than 8 years ago, thanks to these new sequencing techniques. So these bacterial groups are collectively known as the superphylum Pathecibacteria, and they seem to comprise a large fraction of the total known diversity. I will talk a little bit about this bacteria later, but I just wanted to highlight that these groundwater microbial communities harbor a vast amount of species and metabolisms waiting to be discovered. So this is the reason why the number of studies on groundwater microbiota had grown so much in recent years. And although comparably the coastal groundwaters have received less attention, we notice that this growing body of inland groundwater research would be advantageous for us because it provides a solid basis for anticipating the main challenges that the study of the microbiology in coastal aquifers or subterranean estuaries might face. So throughout this talk, I will first present some of the knowledge on aquifer microbiota recently gained from inland groundwater research, and then I will compare it to what is known so far from coastal aquifers based on our review. And finally, I will also present you some preliminary results on a coastal alluvial aquifer close to Barcelona, where we are currently working. I need a little bit of water. Okay, so what are some of these features of groundwater microbial communities that pose challenges for its study? Well, one is that it seems that most of groundwater biomass and activity seems concentrated on aquifer solids. So as you all know, subsurface ecosystems tend to have much higher solid water ratios than surface aquatic environments. So this challenges enormously the sampling and the study of the aquifer microbiome. This is because not only as free-living planktonic cells suspended in groundwater, but also attached onto solid aquifer surfaces. And actually, there have been many studies on inland aquifers showing that most of the microbial biomass or activity is concentrated on these surfaces. So for example, in this study, Gribler and collaborators estimated or quantified the ratio of attached to suspended cells. And they found that it changed from 60 to 1 in a contaminated aquifer to more than 1,600 to 1 in a pristine oligotrophic aquifer. So this means that there are more bacteria in sediments than in groundwater, but also that the physicochemical environment will determine the proportion of bacteria that is attached onto aquifer surfaces. In turn, this will have implications for the functioning of the aquifer microbiome, because there are several studies that have shown that these attached and suspended bacterial communities differ in their species composition. So for example, in this survey of 25 wells in the US, they found a higher proportion of iron and sulfate reducing bacteria in the sediments compared to the groundwater, where they found more methanotrophs. So this means that we may have a really biased or incomplete view of the aquifer microbiome if sampling only groundwater or only the sediments. So for our review, we compiled all the studies that had explored microbial communities in coastal aquifers connected to the sea in the last 20 years. And we found these 36 microbial surveys that are indicated on the map as the orange dots. So we found that none of these studies had actually compared explicitly groundwater and sediment communities, but instead they had focused on either one or the other compartment. So this means that we know nothing or almost nothing about how the species or functions distribute among these two compartments in coastal aquifers. So as I told you before, we are currently working in a coastal alluvial aquifer connected to the Mediterranean Sea, close to Barcelona, as part of the my project, Grammy project, and this project, Medistres 3, led by Albert Folk and Jesus Carrera. So this is an experimental site that was established in 2015 with the construction of a series of piezometers that allow collecting samples along the whole salinity gradient that establishes through the mixing of fresh groundwater that flows seaward in the upper part of the aquifer and seawater intrusion that affects the deeper part. So of course, this is a very simplified scheme of the site. So if you want to know more about the hydrology or the geology, you can check this. So we have performed a series of campaigns in the site, and although we are still processing the data and we have not compared groundwater and sediment communities, microscopic samples of the groundwaters already saw the presence of some particles that are heavily colonized by bacteria, as you can see in this picture. So this means that a large fraction of the bacterial communities may be actually attached to sediments or even to moving particles, and therefore that by sampling just the groundwater, we may be losing a very important microbial component of this site. Some other information that we have obtained from these microscopy observations is that bacteria in this site seem to be much larger and, well, we have more bacteria and they are much larger in the saline groundwater sites compared to the fresh groundwater sites. And you can see that easily in these two pictures of fresh groundwater bacteria at the top and the saline groundwater bacteria at the bottom. So this already suggests that there will be metabolic differences in these two communities. And actually, by measuring leucine incorporation rates, we found the highest bacterial heterotrophic activity in the deeper saline groundwater sites of the system. So this means that there is a large spatial heterogeneity in these microbial properties, which would require spatial samplings, very highly resolved spatial samplings that are usually not very easy to conduct. Well, also, although these microscopy and activity assays are routine in most surface aquatic microbial studies, we found that among our 36 papers, only 4 had applied some microscopy, because most of them had been based on the PCR detection of species. So this means that we are also missing the most basic microbial information of these systems, for which we know almost nothing about the abundances of the bacterial communities, their size, their bulk activity rates, and so on. Okay, and going back to the sediments, in our site, given the difficulty of sampling sediments in this site, an option is to incubate sterile sediments within wells or within piezometers, which is what Bella is doing as part of her master thesis. And she will sample in a few weeks, and we will see which microorganisms have colonized these sediments and whether they differ from those found in the surrounding groundwater. Okay. Another feature of the groundwater microbial communities is that they seem to harbor large numbers of very, very small bacteria, which are called the ultramicrobacteria. So these ultramicrobacteria appear active and dividing in groundwaters, and many of them belong to this superphylum Pathecibacteria that I told you about. So the presence of these small bacteria poses challenges. It's also challenging for different reasons. So first, most groundwater bacteria and aquatic bacteria in general are collected onto 0.2 micron pore size filters. So, and these bacteria can be smaller than that, so we may miss them. Then it's known that some of the PCR-based approaches fail to detect this group. And finally, since they have been just recently discovered, we don't know much about their metabolisms and roles in these systems. So among the studies in coastal aquifers, we found that Pathecibacteria had only been found in some occasions, and in general, they comprise less than 2% of communities. So we might think that they are not important in these systems. However, in our study site, we have been able to identify tiny bacteria labeled as Pathecibacterial cells, and which are actually quite abundant. And we have seen that they occur mostly as free-living cells and are much more abundant in the deep saline portion of our aquifer. So this means, to our knowledge, this is the first direct visual observation of these tiny bacteria in a coastal aquifer, and it might imply that these bacteria are actually important in coastal groundwater areas. But bacteria can also be small because some can miniaturize in response to stress or starvation conditions, such as, for example, the situations of organic carbon, deserved organic carbon scarcity typical of most aquifers. For example, in this study, they found that the exposure of groundwater bacteria to carbon limiting conditions resulted in a significant reduction in their sizes. So this has also implications for our understanding of aquifer microbiota, not only because these miniaturized bacteria will be inactive and therefore not contributing to ecosystem function, but also because it seems that many of them retain the capacity to reactivate and to grow again when the conditions change. So they are considered to comprise what is called a microbial seed bank. And also many of these miniaturized bacteria could be alloctonous inactive bacteria that arrive to the aquifer from hydrologically connected systems like overlying rivers, lakes, or the soils. And of course, this will be much more important probably in coastal groundwaters due to the continuous inoculation species from the fresh groundwater and marine members. But of course, this all will depend on the geology, the geological setting of the site and the permeability of the sediments. And very little has been done about that. So, okay, this groundwater bacteria could thus be either active or inactive. And finally, these aquifer microbial communities have been shown to be metabolically very complex. So they harbor all kinds of metabolisms, most of which are likely unknown. So for that, we rely, since we don't know what these species do, we rely on the sequencing techniques to unveil which might be the metabolisms that hide in these aquifers. So several recent metagenomic studies on inland waters have unveiled an enormous diversity of metabolisms within these systems, which include not only microorganisms with the capacity to degrade dissolved organic carbon using all kinds of electron acceptors, but also microorganisms able to fix inorganic carbon, CO2, using the energy from the oxidation of different reduced compounds such as ammonium, nitrate, or reduced iron and sulfur compounds. So actually this chemoautotrophy seems to be much more important than previously believed, because it's a good strategy to survive in organic carbon limitation conditions. And it seems it might be that these chemoautotrophs are also or provide sources of new dissolved organic carbon for other heterotrophs. So, yes. So in our, among the 36 studies on coastal aquifers, well, in general, we know very little about the microbial metabolisms hidden in these coastal aquifers, because the majority of these studies have been based on the PCR detection of microbial species. So also these studies have provided a very valuable information regarding the main microbial groups that we find or the drivers of these communities. The truth is that we can know little about their function because we don't know what most of they do. So an alternative is to sequence directly functional marker genes so that we can identify specific microbial groups involved in a given metabolic process. So in our review, we try to summarize all the functional marker genes that have been used in inland aquifers to track specific metabolic processes, which are indicated as these color bubbles. So for example, we the AMO8 gene, which codifies for the enzyme ammonia monooxygenase that is involved in the first step of ammonia oxidation to nitrate, or this other gene that converts nitrate to nitrate. So we found that only four articles out of the 36 had sequenced directly specific marker genes, trying to identify the nitrifiers, ammonia oxidizers, or anammox bacteria. So as you can see, they all are involved in the nitrogen cycle. And to show you an example, in this study, Santoro and collaborators explore the diversity and abundance of ammonia oxidizer communities. And they did so by sequencing this AMO8 gene that codifies this first step in the nitrification process. So they found that while the abundances of the ammonia oxidizing bacteria were much lower in the fresh stations compared to the saline ones, there was another group, the ammonia oxidizing archaea, that remained constant across the whole transit. So this was due to different responses of these microbial groups to salinity, which altered the ratio between the two, but with yet unknown implications for the actual ammonia oxidation rates. But more interestingly, they found that this ratio between the two groups moved together with the seasonal displacement of the brackish mixing zone. So which highlights a tight link between coastal hydrology and microbial communities. And also that these dynamic and pronounced salinity gradients present in coastal aquifers might be really strong drivers of these microbial communities. And actually salinity is one of the most important drivers in microbial diversity in general, because it structures communities in nature, defining exactly or determining which species can thrive along these salinity gradients. So for example, in this compilation of global studies from different terrestrial and marine systems, they found that non-saline and saline environments were totally distinguished by their species composition. So in our Mediterranean site, we found very highly diverse and heterogeneous microbial communities that varied largely at the spatial scale, but we could see a clear clustering of these communities following the salinity gradient. As you can see in this ordination graph, where each dot is a community, and you can see that the communities from the freshwater clustered together and were different from those in the brackish groundwater in light blue and then the saline groundwater. So this might imply that any changes in groundwater level or the extent of seawater intrusion will imply large changes in the composition of these communities and in the associated functions. So based on some studies on coastal aquifers and in other systems, it has been suggested that increases in salinity will favor metabolism such as DNRA, sulfate reduction, and overall carbon remineralization, whereas decreasing others like nitrification, denitrification, anammox, and methanogenesis. So this will be due in part to the different responses or tolerances to salinity of the microorganisms involved, but also, of course, to the fact that the availability of the different chemical substrates is also dependent on the mixing of the different water types. But despite this high highly dynamic nature of the coastal aquifers, what we found was that most of these studies sampled only in one occasion, which will thus provide just a snapshot of a situation that will likely change in the future, or seasonally, or even daily, due to tides, storms, waves, groundwater level, and so on. So future studies should better explore this temporal variation. Okay, and now moving to the marine side of the study. So in our review, we also wanted to compile what was known about the responses of marine bacterioplankton communities to SGD. So we focus on marine bacteria because they are key players in marine ecosystems, because they have been much less studied with regard to their responses to submarine groundwater discharge than phytoplankton or other organisms. However, if you are interested in the SGD effects on marine biota in general, you can refer to this very nice review by Lecker and Maki. So now we know that bacteria control the flow and fate of carbon and other nutrients in the ocean. And for example, they can take up a large fraction of the dissolved organic carbon and either respire it to CO2 or incorporate it into their biomass so that it becomes available to higher trophic levels, because there are many microorganisms that feed on bacteria. So together with phytoplankton, bacteria are at the base of marine food webs and ecosystems. So the effects of discharge on marine bacterioplankton could be direct due to decreases in salinity, changes in temperature, or nutrient or carbon inputs, but also indirect due to effects on other microorganisms affecting bacteria, such as phytoplankton, bacterial predators, or viruses. But in any case, in these two cases, the composition of the bacterial communities will play a large role, because we know that different species react differently to changes in conditions or can be differentially controlled by these biotic factors. So we could only find 6 studies that had linked the responses of natural bacterial communities to SGD, and are indicated here at this, like these blue dots. So for example, in this case, Carlson and Wigner explored the activity and abundance of bacteria in coastal sites of Hawaii, and they found lower bacterial growth efficiency in situations of higher submarine groundwater discharge. So if the bacterial growth efficiency is lower, it means that this bacteria will likely channel a small fraction of the dissolved organic carbon that they consume to higher trophic levels, and they could instead be acting as sources of CO2 to the atmosphere. So this would have implications for the carbon cycling. And on the other hand, other studies doing experimental additions of groundwater to the sea, to marine communities, have found either growth of photosynthetic bacteria in the case of the Mediterranean Sea, but not in the case of Hawaii or California, where other groups were favored. So this means that these responses will not only depend on the taxonomic composition of these communities, but also on the chemical composition of the groundwater probably, and on many other local or biotic factors. And finally, submarine groundwater discharge could also influence marine bacterial communities through the direct transport of groundwater microorganisms. So yes, this connectivity between the aquifer and the marine environment has been mostly studied in terms of the transport of terrestrial pathogenic or fecal bacteria, but to our knowledge, very few studies have actually suggested a role of submarine groundwater discharge as a conveyor of natural microbial diversity to the marine environment. So an example of that is this study by Lee and collaborators, where they found that submarine groundwater discharge together with tidal fluctuation determine the periodic occurrence of bacteria in coastal bacterioplankton assemblages, like these beta proteobacteria indicated here in yellow, whereas in high tide, other typical marine bacteria dominated. So this also indicates a tight link between the composition of bacterial communities in the coast and submarine groundwater discharge. However, in our Mediterranean aquifer, we found much lower connectivity, as you can see in this graph, where the black indicates the proportion of species that were present in both the aquifer and the sea. And as you can see, they represented a very small fraction of the communities. So this might be due to the microtidal regimen of the Mediterranean Sea, but also to the much lower permeability in these sediments compared to the previous study, which mostly comprised volcanic fracture rocks. So depending on the geology, it's probable that the environmental gradients that the microbial communities experience will be very different, as well as the extent of the discharge of groundwater diversity to the ocean. But in general, very few studies have explored both the communities from the aquifer and the seawater, so we know very little about this potential dispersal of groundwater microbes into the sea. So far, what we have seen is that although the study of coastal aquifers seems quite in its infancy, we can learn from previous research on inland aquifers to try to anticipate the challenges that this field might face. So what we have learned so far is that these aquifers hide very diverse and heterogeneous communities that vary largely even at the small spatial scales, that salinity is a major driver, and that hydrology make these systems more dynamic than inland groundwaters, that depending on the geology, the potential connectivity with surface or seawater might be important, and then there is very little knowledge on the effects of submarine groundwater discharge on bacterial communities, but that some studies have found effects in bulk bacterial functions or cyanobacterial growth. So as you can see, there is a lot to do. So in our review, we propose some of the research lines that this field should tackle in order to advance our knowledge. And for example, we believe that the studies should try to better address the spatiotemporal variability in microbial communities, as well as this connectivity with surrounding ecosystems. We should also try to consider attached and the suspended bacterial communities, as well as the poorly explored ultramicrobacteria, to have a more complete view of the aquifer microbiome. And finally, if our aim is to try to understand what are the processes that take place in this aquifer marine continuum and how these microbial processes might influence or determine the chemical fluxes associated to submarine groundwater discharge, studies should move towards studying functions, not only species composition, because with the ultimate aim to try to link this microbial information with actual metabolic rates. And actually, this is one of the most challenging questions in microbial ecology due to this complexity and diversity within bacterial communities. So for example, the measurements of bulk activity rates, such as heterotrophic activity, gas production, extracellular enzyme activity, often treat microbial communities as a black box as a whole. However, we know that different species may have different environmental tolerances or preferences or activity rates, so it might be very difficult to predict how these potential rates will change upon changes in environmental conditions. On the other hand, all these genomic tools have allowed to open this black box. And I think this will be of great use, because just knowing which of the metabolisms prevail in your system will give you an idea of what may be the main processes operating. So for example, even though we know that both anamox and denitrification are responsible for removing nitrogen from the system, depending on which one dominates, you may expect a different outcome, a different response upon salinity changes, or there will be different products, different chemical elements produced, for example. In any case, still the presence of a gene that does not imply that the associated process are occurring at a given rate. So this link is still not straightforward. And finally, there are other less commonly applied techniques, such as this, for example, stable isotope probing of microbial DNA that allow linking or allow identifying specific microorganisms involved in the use of a given substrate labeled with stable isotopes. So, for example, this has allowed to unveil which microbes are responsible of autotrophic fixation of CO2 or in the degradation of plant material in groundwaters. So in my opinion, we just should move to a combination of techniques that together the most complete picture of these microbial communities in coastal aquifers. But of course, we cannot, we need to accompany this with a good hydrogeological knowledge of the systems, of the oceanographic setting and the biogeochemical processes taking place. So there is a need to foster interdisciplinary collaborations. So, well, that's it. This work is mostly part of my project GRAMI and resulted from our initial collaboration a few years ago with Valentin, Jordi and their group, impulsed by Pep Gasol and myself. And some of the data shown have also been processed by Elena and Lara, who did their master thesis with these first samples from the Argentina aquifer. But we are currently many people working together as part of different projects related to submarine groundwater discharge in different systems in Spain. So this group is currently growing, and hopefully soon we'll be able to tell you a little bit more about this poorly explored microbial dimension of submarine groundwater discharge. So with this, I finish. I don't know if I have been too fast, and I hope it has not been too microbial for non-microbial people. But I thank you for your attention, and I will be happy to take any questions.
Thank you so much, Clara. It was a very informative talk for SGD people. Some people know more than me. Thank you so much. It was exactly the right size and the right pace.
Okay. Yeah, I don't know if it's okay.
Okay, so I'm gonna open now to the audience for questions. So please unmute yourself, and yeah, Dini's first. Dini, go ahead.
Hi, thank you, Clara, for your talk. It's really great. Yeah, so I have one question related to your current project. So you mentioned that you found the shared species between all the well samples. And can you share whether this this shared species, like, for example, which group, like whether they belongs to specific metabolism group, or are they like more general bacteria that you can find anywhere, such as like Pseudomonas also?
Yeah, actually, if I remember correctly, so I found both types. So there were some were typical marine bacteria that we found in the aquifer, and the others were other groups common groups. I think there were some also things like that were found in the aquifer. So we don't know. Well, the typical marine bacteria, I guess it's just by dispersal somehow with waves or storms or things like that we can detect their presence in very low abundance in the aquifer. And regarding the others, we don't know what they do, but they seem to be quite, so they are some of the few bacterial species that we find in the aquifer that are quite widely distributed, because we find that in each site we have quite unique bacterial communities. So there is not a lot of dispersal even within the aquifer, but there are some microorganisms that appear to be more tolerant to the gradient. And I think those were the ones that we could also detect in the sea. If I remember correctly, I think they were some gamma proteobacteria and some rhodobacteriales, I think. And we have also, and actually we have managed to isolate some of those bacteria from fresh groundwater growing in marine media. So I guess that there are some things that actually can grow when arriving to the sea.
Mm-hmm.
But I don't know anything about their metabolisms, but we might try to sequence them when we get them in isolation.
Uh-huh. Okay, and this is based on sampling from different regime, like how many sampling session, like it's between different tidal cycle or different seasons, or
Yeah, no, this data that I saw was just during summer. So just one day of sampling. So one snapshot. Of course, during summer, there will be less connectivity even, so less discharge in general. But now we are doing, so we just conducted one sampling in winter. So high flow, let's say, after some precipitation, and then we will do other in the summer, and we haven't been able to look at this data. So everything is very preliminary. We haven't had the time so to look carefully on them. But I think the story will change, I expect.
Great, thank you.
Thank you, Dini.
Any other questions? Buchao, go ahead and ask.
Natasha,
Hey.
that's Clara. Hi.
Hello.
Clara, hi. I have a question. We did some work years ago along the Eastern China coast in the coastal aquifer. We did a time series.
Okay.
24 time series, and one of my colleagues did. He looked at the community of bacteria and archaea, and a lot of microbiology stuff. Based on his data, I remember I saw his data years ago, and based on his data, I thought we can see the time series pattern that during the rising tide, the community is more like the oceanic community.
Yeah, that
but
makes
during
sense.
during low tide is more like terrestrial.
Terrestrial. That is very nice. Is that published? Have you published that? Because I don't think I have seen it. Oh, now I don't hear anything.
Yeah, maybe something happened with the connection on his end.
Okay.
So, Buchao, are you still there?
No, I think we.
Okay, maybe we lost him.
So that is very interesting. I don't think we will see that in our aquifer because in the Mediterranean we don't have big tides. So we see that there is connectivity very close to the shore, but within the marine, the groundwater, the saline groundwater communities are really, really different from those in the sea. So there is not a lot of transfer. Well, the water resistance time is very, very high. So the communities are really groundwater communities, very different.
All right. Any other questions, guys? So when you talk about, guys, and I'm one of those people when we refer to this as the biological stuff, because I have very limited knowledge in that. But when you talk about now, I'm thinking about tidal changes in this microbial community, is it, I know it's a very basic question, but is it because of transport with the water of the communities? Or I've heard that they can change their genes, and depending on the conditions, they could sort of do different things. The same bacteria, but changing their genes, and now they're doing different things because the conditions are favorable for a certain pathway of biogeochemical changes.
So in our case, I wanted to explore that, and that is why I compare whether we have many shared species, so they just change their abundances, but they are there everywhere, or whether they were in each site and in the sea versus the aquifer. And what we see is that most of them are very different. So it doesn't seem that it's a species that thrive throughout the aquifer and they can just adapt to the different conditions, but it seems that they are really different. However, I would expect that in more permeable sites where you can have more transport of things with waves and things like that, many of them, there are some that are quite versatile, so they can really tolerate salinity changes and things like that, like the things that we retrieve in our cultures, because these were in fresh groundwater, but they can grow in marine conditions. So it means that they tolerate, or either they are resisting alive in the groundwater and they can grow later. But most of the things that we see, they are really, they seem to be really adapted to the specific conditions that we have in our sites. Of course, the site salinity, we have very pronounced gradients in oxygen, in pH, and in many nutrients. So the conditions seem to be very, very different. And also, apparently, there are many microniches in the porous media. So within pores and within sand grains, there might be smaller concentrations of the sort of oxygen or things like that. So we don't really have an idea of the actual heterogeneity of these communities at the micro scale.
Okay, so just a follow-up question. So based on that, and I know it's difficult to answer, but based on your experience, which is the parameter that you think is the driver of this community changes and abundances?
In here, I would say salinity is really important because it depends on their tolerance. So if they cannot cope with this osmotic stress, they will die. There are some things that are more tolerant, but in general, this other paper that I saw, it shows that salinity is one of the most strong drivers of communities overall, because even more strong than pH and dissolved oxygen and so on. But of course, within this was across ecosystems. Within this site, we tried to quantify the relative role of different drivers, and we had salinity as the most important one, but also, for example, nitrate was also important, but you don't know to which extent this is just because fresh water carries more nitrate or because bacteria use the nitrate and so on. And oxygen, of course, will determine the metabolisms that can be. But when you look at the whole community, salinity really is the thing that makes your communities cluster more clearly. But then if you go at the level at the species level and you see whether this species thrives here or here or here, there will be other drivers that are explaining the presence of a species in that range of conditions.
So the salinity basically impacts directly their cells, or is it the salt that sort of kills them?
Yeah,
how it
they
works?
have to be able to control the osmosis. So if not, they will die. They have to be adapted to cope with high salinity outside or inside.
Okay.
So, but this is so usually that is why explains the segregation between freshwater and marine communities. But we know that there are different mechanisms to cope with excess of salt. So there are some organisms that can do that and can adapt to different salinity levels. But in general, it has driven also the evolution of different species adapted to one thing or the other. But then we see things that are able to persist, like these things that grow. So some of the bacteria are quite amazing at persisting for very long periods of time, inactive but alive. So, in unfavorable conditions. So it seems that is why one of the aims of my project was to see to which extent these coastal aquifers might be sources of microbial diversity for the ocean. So that is why we try to do these things. So we still don't have an answer, but certainly there will be things that are able to grow when they arrive, or even things that don't grow when arriving to the ocean, but they can be a source of food for other bacterial predators, perhaps, and thus constituting a source of allocturnous organic carbon for the coastal environment.
Thank you. Fascinating. More questions, guys?
If there is no one else that want to ask, I have one follow-up question.
Okay.
So I suddenly remember this one case that we have that when Natasha asked about these different genes that may be activated during certain conditions. So for example, in our study right now, we found based on the sequencing, there are a lot of sulfate. Like if you see their names are like disulfovibrio and so on, and so there must be sulfate reducing bacteria based on the sequencing. However, when we do the PCR, so we test PCR for like different processes, including denitrification and also sulfate reducing processes. But based on this PCR, it seems like the denitrification gene copies is really high, while sulfate reduction gene copies is pretty low, even though based on sequencing, it's like there are high abundance of sulfate reducing bacteria in that sample. So do you have any comments about that?
Well, it could be also, we have, as you may know, this problem with the 16S, that they can have multiple copies, some of the microorganisms. So the relative abundances based on this marker gene of taxonomy composition may not be exact. And if you, I don't know how many copies can have of the other functional genes, but perhaps if these sulfate reducing bacteria are very large and they have many copies of the 16S, this could be overestimating their numbers, whereas they might not be so active and they not be contributing to that function in particular. So I think this is because you did a qPCR of the DNA, no? So it's genes, it's not transcripts.
Yes.
Yeah. So I guess that you have a different picture when you have when you sequence the whole communities, and it's all is relative. Also, it's also dependent on the sequencing depth that you use, no? And if you are only targeting a specific functional gene, you will amplify all that you have there, but not with respect to the other things. So I don't know if that could be an explanation, but I will. And also all these things, all these primer-based approaches also have some biases against some groups.
Right.
So so perhaps either the 16S primers that you use were not capturing some of the other denitrifier groups that you had, or vice versa. Some of the specific marker genes are not super good for those groups, because since we don't know most of these species that hide in these aquifers, and all those primers are based on the available sequences, then it is known that some of them fail to capture some of the groups. So perhaps this abundance bias is not complete coverage of the diversity that exists within a group.
Yeah, I think that makes sense. And I mean, we also want to do like transcriptomic with RNA, so that might answer some of the questions.
Yeah, for sure. Yes, I haven't almost talked about RNA, but that could also give many answers about what is active in that a particular moment. So so nice. So you are working on that, and you will have results sometime.
Yeah. So hopefully next month we will have results.
Okay.
Okay,
Good.
yeah.
So good
Thanks.
luck with that. Yeah, I
Yeah,
would like to
thanks.
move towards that, towards the qPCR detection of a specific functional genes. In our case, the nitrogen cycle seems to be very important because there is a lot of nitrates derived from agriculture. So it would be nice.
Yeah.
Thank you, Dean.
Thanks.
Thank you, Dini. Guys, any more questions? All right. This was really fascinating talk and very educative. And like last few talks we had, I hope this helps the community to, learn more things from the comfort of their computer. I want to thank Clara for giving such an interesting talk, and everybody attending. As always, Isaac is going to put this on the YouTube channel, so if you have missed something, you can always go back and check it. And we're looking forward to more publications on the topic of microbial communities at the interface of groundwater, surface water, ocean. So thank you everybody again for attending, and specifically Clara. Thank you guys.
Thank you, Natasha. Bye-bye.
Bye.